Welcome to all. I am Dr. Dharmendra, Assistant Professor of Botany, presenting this session. In this session, we will going to discuss about reproduction types present in Oedogonium plant. Before that, we will recall the memory of a previous class. In the previous class, we discussed the occurrence of Oedogonium thallus, structure of Oedogonium filament and the cell, and the method of cell division present in the Oedogonium. So in this session, we are going to learn the objectives, that is studying of vegetative reproduction, objective 2, studying of asexual reproduction, and studying of sexual reproduction present in Macrandrous species. We will discuss one by one. By this, at the end of this session, you will going to learn the knowledge of reproduction types present in the Oedogonium plant. So coming to the reproduction, so Oedogonium, the plant reproduces by three methods, that is vegetative reproduction, asexual reproduction and sexual reproduction. So these three types of reproduction present in the Oedogonium. First one is vegetative reproduction, second one is asexual reproduction, and third one is sexual reproduction. So look at this, the first one, that is vegetative reproduction. So vegetative reproduction is the most common method of reproduction, and this reproduction takes place by means of the fragmentation. So fragmentation means it is nothing but the breakdown of the filament into several pieces. So this process what we call fragmentation, and each segment, that is called fragment, it has the capacity to develop into the new individual plant. Why fragmentation occurs? The fragmentation occurs due to these reasons. The first one, that is dying out of some cells in the filament. So filament, that is an elongated structure, it consists of so many cells. So here and there some cells undergoes death. So because of this dying out of the intercalary cells, the filament gets separated, and it becomes fragments, and the fragment is separated and which developed into the new filament. Then second one is through mechanical injury. So mechanical injury, it is mainly due to water current or by the consumers or by the activity of human beings, whatever it may be. Then formation of spores in the filament. Again, the cap cells which is present in the Oedogonium filament, that is responsible for the production of the spores, akinete spores, asexual spores. And because of this formation of the asexual spores, because of the presence of the spores, the vegetative cells, they are disconnected from each other. By this, they are separated from each other and they develop into new fragments. So these three reasons are responsible for the fragmentation takes place in the Oedogonium. So that's why Oedogonium, the fragmentation is the common method of vegetative reproduction, or it is the common method of reproduction. Then coming to the asexual reproduction. So asexual reproduction takes place by means of formation of asexual spores. So asexual spores formation takes place here. So there are two types of asexual spores. One is akinetes and second one is zoospores. So these are the two types of asexual spores. Akinetes and zoospores formation takes place. So we'll discuss one by one. The first one is akinete formation. So akinetes are the resting spores that formed during unfavorable condition. Look at this diagram. So this is the Oedogonium filament that consists of a chain of akinetes, and these akinetes are rounded or oval-shaped cells. We will discuss the formation of akinetes. So during unfavorable condition, the plant body produces akinetes. So because that's why they are also called resting spores. So these akinetes are found usually in the chain of 10 to 40 in flattened cells. And these akinetes resembling oogonia, because they are oval in shape, that's why they looks like oogonia. So usually akinetes are arranged in 10 to 40 in flattened cells. So akinetes are formed only during unfavorable condition. So because of these akinets, they are thick-walled. The wall is very thick and the dish brown, more or less rounded in structure, and the protoplasm is rich in starch and the dish orange oil. So again, akinets are the resting spores made up of thick-walled layer, and this thick-walled layer encloses the large quantity of reserve food material. The reserve food material is nothing but the starch. Okay, and this akinets overall color of akinets looks like reddish orange in color. Okay, because of the presence of reddish orange oil in the protoplasm, it looks like reddish in color. So these akinets, because of the thick wall, they resist the unfavorable condition. Whenever the conditions are favorable, so these akinets germinate directly and they give rise to new filaments. So that's why akinets are also called resting spores, because they are formed during unfavorable condition and they withstand the unfavorable condition, and during favorable season these akinets germination takes place. Okay. The second type of asexual reproduction, that is zoospore formation. You see the zoospores, they are formed in a species of Oedogonium. So these zoospores are the motile spores. Okay. So it is the most common method of asexual reproduction. Zoospore formation also one of the common method of asexual reproduction. So these zoospores production takes place and it multiplies the number of filaments. Okay. And the more important is the formation of zoospore is depending upon the presence of carbon dioxide in the surrounding environment. So the carbon dioxide concentration that influences the zoospore formation in the filament. Okay, so this is one of the important factors. How this zoospore formation takes place. Again, any one of the capsule in the filament that produces zoospore. So that cap cell acts as a mother of the zoospore. That's why those cap cells, that is what we call zoosporangium. So zoosporangium is the mother cell of the zoospore. It is nothing but a cap cell. So this cap cell involves in the production of zoospore. So those cells, what we call zoosporangium. So the zoosporangium produces only one zoospore. So only one zoospore formed takes place inside a single cell. Okay, a zoosporangium gives rise to single zoospore, and this spore is oval in shaped with multiciliates. Okay. So what happens during the formation of zoospore? The protoplast produces large amount of the reserve food material. This is the first stage. The protoplast accumulates large quantity of reserve food material, and this protoplast is present as a lining layer, and this protoplast separated from the wall, that means cell wall. Okay, the protoplast produces large quantity of reserve food material and gets separated from the cell wall. Then what happens? After the separation, the protoplast becomes a rounded body. Okay, it becomes rounded body and it develops a semicircular or lens-shaped colorless area at the anterior end. So the protoplast gets separated from the cell wall and it becomes oval or rounded in shape. And at the anterior position, it develops an empty space. That means colorless area. So this empty or colorless area, what we call bald spot. Okay, bald spot. Appearance of bald spot takes place after separation and after forming the circular form. Then below the bald spot, the basal granules of the blepharoplast forms and they are arranged in a complete ring-like manner. Okay, bald spot is present at the anterior portion, that is a colorless area. So below that colorless area, these blepharoplast, that means the basal granules, they are arranged around this bald spot around as a ring-like manner. They are arranging in a ring-like manner. So these blepharoplasts are very important because these blepharoplasts that gives rise to the flagellum or the cilia. From a basal granule, single cilia appearance takes place. Okay, look at this point. From each granule, a single flagellum or cilia rises. So this kind of arrangement of cilia, what we call stephanocont. So stephanocont means it is a ring of cilia arranged around the bald spot. So these points are very important. That means the words bald spot, blepharoplast, and stephanocont. Okay. Now what happens? The juice spore is matured. After the matured juice spore is a larger in shape. It is a pear-shaped structure with full of reserve food content, and it consists a ring of cilia at its anterior narrow end, that is below the bald spot. Now the juice spore is matured. The juice spore is ready for liberation. Okay, how the juice spore is liberated? Look at this point. The upper wall of the cell. So we have the juice parenchyma again above the vegetative cell. There is a apical cap is present at the upper side. So this upper wall of this juice parenchyma splits transversely at the junction of apical cap. Okay. So it splits, this is the apical cap. At this junction, it splits transversely and it forms a pore. Okay. So the juice pore comes outside from the juice parenchyma with a vesicule or with a mucilaginous sheath. Okay. So the mucilaginous sheath that brings about the juice pore come towards the outside through that aperture formed on the transverse wall. So and this mucilaginous sheath later dissolved and the juice pore is liberated in water, and this juice pore swims for some time and it withdraws the cilia and it attaches to the substratum with that bald spot. The bald spot area attaches to the substratum and the germination of juice pore takes place by means of the division and it gives rise to the new filament. So this is the process and liberation of formation and liberation of juice pore. I will show the diagram. Look at this diagram, this side, that is figure 3.74. So this is a juice parenchyma cell. Look at this, the protoplast accumulates the reserve food material and it withdraws its contents from the cell wall. It becomes rounded in body. Okay. So now again it accumulates large quantity of reserve food material. Reserve food material takes place here. So this reserve food material takes place and it becomes the circular form. Now we can see the juice pore formation takes place here. The bald spot, look at this diagram. This is the bald spot. So below that bald spot, a ring of cilia present. So this arrangement of this type of cilia, what we call the stephanocont. The juice pore comes out by breaking the transverse wall present at the junction of apical cap. At this point, it breaks open. So the juice pore comes along with a mucilaginous sheath, and this mucilaginous sheath is dissolved and juice pore is attached to the substratum with the help of this bald spot. It withdraws its flagella and this bald spot becomes apteron or holdfast and the juice pore germinates and it gives rise to new filament. So this is the complete process of juice pore formation. Okay. By this, asexual reproduction takes place by means of formation of akinets and formation of juice pores. Then coming to the sexual reproduction, here Oedogonium, the sexual reproduction is of oogamous type. That means advanced type of sexual reproduction. Based on the development of antheridium, the species are classified into two types. That is Macrandrous species and Nanandrous species. It is mainly based on the development of antheridium. Okay, so one is Macrandrous species and Nanandrous species. So we will discuss the sexual reproduction in each species one by one. Okay, first we will discuss the sexual reproduction in Macrandrous species. So what do you mean by Macrandrous species? So Macrandrous species here, the antheridium development takes place on the normal sized filament. Okay, the normal sized filament that gives rise to antheridium. So those species are called Macrandrous species. Again, they are the dioecious forms or monoecious forms. Dioecious forms means the antheridial filament is separate, oogonial filament is separate. Both antheridia and oogonia, they born on the separate filaments, that is dioecious species. In monoecious species means antheridium and oogonium, they born on the same filament. Okay, that is monoecious species. So macrandrous species, that is nothing but the antheridia development on the normal sized filament. So they may be dioecious form or they may be monoecious form. Okay. How this antheridium development takes place? We will discuss the steps. You see, the first step, any one of the vegetative cell is exclusively cap cell. So cap cells are very important because they give rise to juice parenchyma and the gametangial cells. So any one of the vegetative cell, that is cap cell, acts as antheridial mother cell. Okay. Any one of the cap cell acts as antheridial mother cell. Then what happens? This antheridial mother cell undergoes repeated divisions. So transverse divisions occurring on this antheridial mother cell. And this transverse division results in the formation of a row of flat, short, cylindrical, disc-shaped cells. So these short cells, what we call antheridial cells. So one of the capsule which undergoes division, that is transverse division, occurs, and this transverse division results in the formation of a small sized rectangular shaped cells. So these small sized rectangular cylindrical shaped cells, what we call antheridial cells. So the number of antheridial cells in a row, that varies from 2 to 40 based upon the species. So a single vegetative cell that is antheridial mother cell, that may give rise to 2 antheridial cells or maximum of 40 antheridial cells. Okay. The number of antheridial cells ranges from species to species. First, what happens? One of the cap cell acts as antheridial mother cell. The protoplast of antheridial mother cell undergoes division. So the repeated division occurs. This repeated division results in the formation of short disc-shaped cells. And these disc-shaped cells, what we call antheridial cells. The number of the antheridial cells varies from 2 to 40. Okay. Then what happens? The protoplast of each antheridium cell, again that undergoes division, and it gives rise to 2 masses of protoplasm. Okay. The protoplasm of each antheridium cell undergoes division, and it gives rise to 2 masses of protoplasm. Each mass is developed into an antherozoid or a spermatozoid. Okay. So each sperm, it looks like the zoospore, but it is smaller than a zoospore. It is usually oval in shape with a bald spot towards apex and a crown of cilia present below the bald spot. It is exactly morphologically resembling with zoospore, but the size of antherozoid is smaller than the zoospore. Again, it is a uninucleated spore. The sperm consists of a single nucleus, cytoplasm with less chlorophyll and reservoir material. Like the zoospore, the antherozoids are also liberated from the antheridial cell by a pore appeared at the junction of apical cap near the transverse wall. So these antherozoids comes out with the thin vesicle or mucilaginous sheath. So then it liberates from the mucilaginous sheath and it swims in water and it reaches the oogonial cell. Okay. So this is the diagram explains the formation of antherozoid. Look at this. This is the cap cell, acts as antheridium mother cell. So the protoplasm of this cap cell undergoes division. It results in the formation of the cells. So this is saying antheridium. The division continues and it forms a small flat shaped cells, and these small flat cylindrical shaped cells are called antheridial cells. And the nucleus or protoplasm of this antheridium cell undergoes division and it gives rise to 2 masses of protoplasm. Look at here, 1, 2, 1, 2. So each mass is developed into a single antherozoid. So a single antheridium cell consists two spermatozoids, or a single antheridium cell gives rise to two spermatozoids, and these spermatozoids are liberated by the pore appeared on the junction of this transverse wall through this vesicle. So you look at the structure of anthozoid. Again, they are oval in shape with a bald spot covered by a ring of cilia, and it is a uninucleate spore which is exactly resembling zoospore, but it is smaller than zoospores. Okay, so this is the development of anthozoids in Macrandrous species. So coming to the development of Oedogonium in Macrandrous species. See, the development of Oedogonium is common for both the Macrandrous species and Anandrous species. Okay, so the Oedogonium development is common for both Macrandrous species and Anandrous species. That means again any one of the cap cell of the filament that acts as oogonial mother cell. So this oogonial mother cell accumulates large quantity of food material, and because of this accumulation it bulged in structure and it undergoes a division. So the division is unequal in nature. It gives rise to two types of cells, that is the upper cell is large and oval in shape and the lower cell is narrow in shape. So this lower cell what we call subflutary cell, the upper cell what we call oogonial cell. Now the protoplasm is completely rounded in shape, and this protoplasm encloses a single nucleus with large quantity of food material, and at the maturity, the just below the apical band junction, that is on the transverse wall, there is a pore formation takes place. So through this pore the anthozoids enter inside and the fertilization takes place. So this we will discuss in Anandrous species. I will explain the development of Oedogonium in detail in Anandrous species sexual reproduction, because both the species the development of Oedogonium is similar and same kind of development takes place. Okay. So in this session we discussed the vegetative reproduction and asexual reproduction by echinid formation and zoospore formation, and sexual reproduction, that is the development of anthozoids in Macrandrous species and development of Oedogonium in Macrandrous species. Okay, so these are the further references, and you can expect these type of questions, multiple choice questions from this session. Okay, so the formation of zoospore is initiated by, there are four options, that is age of filament, amount of water in pond, amount of carbon dioxide in surrounding water, amount of oxygen in surrounding water. So the correct answer is carbon dioxide concentration in surrounding water. Then in zoospore, the ring of arrangement of cilia is called bald spot, blepharoplast, stephanocont, basal granule. So the correct option is option C, stephanocont. Characteristic feature of Macrandrous type of sexual reproduction is presence of anandrium, option B, development of Oedogonium on normal filament, development of antheridia on normal filament, and both B and C. So the correct answer is option D, both B and C, that is Oedogonium and antheridium development takes place on the normal sized filament. Then basal cell present below the Oedogonium is a subflutary cell, basal cell, oogonial cell, all the above. So the correct answer is called subflutary cell. Then the number of antheridia in a row ranges from 1 to 40, 2 to 50, 2 to 40, 3 to 40. So the correct answer is 2 to 40. By this, we completed the session of Oedogonium reproduction. Thank you.